I have a positive terminal here because I'm using electricity here. This is the magic word here. So these are your keywords. So one mark for preparing the wells and loading them. The second mark is for the gene probe. So read the question carefully. It's a 5-mark question. You have 10 minutes to answer this question. So think carefully and write carefully. Pause the video now. So what we're gonna do here is start by, let me just draw. This is how gel electrophoresis looks. And I start at the negative terminal and I have a positive terminal here because I'm using electricity. I have different wells. I call these wells. So this is the keyword. These are called wells in which you are going to put the DNA samples that you want to compare in. So you would put the DNA samples here, DNA sample 1, DNA sample 2, DNA sample 3, DNA sample 4, and you would put Agarose gel on this whole place. This is a special gel used for electrophoresis that conducts electricity. So you would put Agarose gel and then you would start the electric current and allow electricity to flow through this whole apparatus. Actually, before you start the current, you want to tag these DNA profiles. Why? So that later when you look at them with ultraviolet light, you can see them easily. So you're tagging them so you can see them. All right? And there are a lot of tags, indicators. I like to use methylene blue. All right? So you tag these things, the DNA fragments, and then you start the electric current. The electric current starts and all of the DNA, because it is negatively charged. So you have to know this piece of information. DNA fragments are negatively charged. So because they are negatively charged, they start moving towards the positively charged terminals. They get attracted or pulled by this. And the shorter the fragment, the smaller the fragment, the more it moves towards the positive terminal. So for example, let's say we have a 1 centimeter fragment here, a 5 centimeter here, and a 10 centimeter here. So when you start the electricity, the 1 centimeter is going to reach this point here. The 5 centimeter is going to reach this point. The 10 centimeter is going to reach this point. Why? Because it's very heavy. It's difficult to make it reach far. So the positive terminal pulls, then all of them are negatively charged. It pulls the negatively charged fragments towards it, and the shortest one reaches further. So this separates the bands or the profiles. We call them bands or profiles. So these bands are separated based on size. All of them are negatively charged, but the size is what differs. All right, and this distribution of bands, you will see something like this. The distribution of bands is the profile of this particular DNA. It tells you what this DNA is made of. Let's have a look at our answers and how I explain this answer or this concept in an answer. It's up to you if you want to write them as bullet points, as 1, 2, 3, 4, 5. I like to write it as a paragraph. As long as you mention 5 points, you will be fine. So start by preparing wells. So first you have to mention the wells, that you prepare wells and you load them with the DNA sample or the DNA fragments and Agarose gel. Then the second step is to add a gene probe and preferably mention one. For example, methylene blue. All right, to tag the DNA fragments. Tag meaning to add like an indicator to the DNA fragment so that it is visible. Apply a current through the gel. When the current runs through the gel, it causes the negatively charged DNA fragments. You have to mention that the DNA fragments are negatively charged and they move towards the positive terminal, which is called the anode. So these are your keywords. So 1 mark for preparing the wells and loading them. The second mark is for the gene probe and tagging the fragments. The third mark is for mentioning that the DNA fragments are negatively charged and they move towards the anode. And then you have to mention the shorter the fragment, the further it moves. So I like to write it this way. You can say the shorter fragments move further. The shorter the fragments, the further it moves towards the anode. This creates multiple fragments on multiple levels based on their size. Compare the DNA profile. So after I have my profiles, what do I do? How do I know if this is a Black Panther or not? So I have a profile. One of these is definitely a Black Panther. I know I got this from a real Black Panther. And now I compare this with all these samples that I got from somewhere else that I don't know. Are they Black Panther? Are they not? And this is what I'm trying to find. So if there is a match in the fragments or the profile, they exactly line up, it is a Black Panther. If they don't line up, it is not a Black Panther. So this is, I explain this by saying compare. This is the magic word here. Compare the DNA profiles, we call them DNA profiles, or you can say compare the bands with the profile of a confirmed Black Panther DNA. If the profile matches, this is also the magic word, then the DNA is from a Black Panther. So you want to say compare and look for a match. Whatever matches is the DNA from a Black Panther. So this is my 5th mark, comparing. And my 4th mark is the shorter the fragments, the further it moves. So this is how you answer a question about gel electrophoresis. Sometimes they don't tell you about PCR. So you have to mention that first you have to do PCR on the DNA and then start by doing electrophoresis. Because electrophoresis without PCR means you don't have enough samples. First you do PCR and then you do electrophoresis. In this question, they already mentioned that PCR was done. So I start from the beginning of gel electrophoresis. I hope that's clear.